Hello, can I ask everybody to take the seats? We will have the last speaker is starting from a different country. We better keep our time. So we already hear a lot of great talks from patient advocates, also from researchers and biotech thought leaders. So our last section of today's event is advancing ALS research. ALS is a neurodegenerative disease. The full name is amyotrophic lateral sclerosis. So it's affecting the motor neurons in the human bodies. It hits usually in the late stage in people's life. And today we have a few influencers in the field. We have 5 influencers in the field to discuss the breakthroughs in mechanism and therapies. So before starting the section, I'd like to play a short video from a friend of our company, and also who is one of my most admired ALS fighter. Close the light. So his name is Jaime Lafita. So a native of Bilbao in Spain. Jaime was diagnosed with ALS in 2016. In response, he founded the Della Candela Foundation in 2019 to raise awareness and funds for ALS research. The Della Candela is the Spanish phrase with the meaning of fighting hard against ALS. Demonstrating the remarkable resilience, Jaime has undertaken significant challenges, including a 600-mile bike ride from Spain to Brussels, Belgium in 2021, where he was received by the President of European Parliament on World ALS Day. In April 2024, he completed a 9-day, 400-mile tandem bicycle journey across Death Valley in the United States. I was honored to join his Death Valley ride, leading a Chinese cyclist team. After the grueling 400 journey, and we ended our ride in front of El Capitan, which is the symbol for conquer the most impossible task. And then we celebrated our accomplishment with the leadership team from ALS Network, and where I met the leadership. Sherry and Cheryl is today here as joining us. So I consider this a small reunion. And through this endeavor, Jaime continues to inspire and mobilize the effort towards finding a cure for ALS. And this is a video he shot for us.
Hi, everyone. Hi, Nayanwei. Hi, iXCell Biotech team, and thank you for thinking about us for this video. This is Jaime from Della Candela in Spain, fighting for a world without ALS. We had the pleasure to visit your company and Don Cleveland's lab when we joined forces for the Death Valley Challenge, as a metaphor for the valley of death that research projects need to cross to become a drug in the market. We wanted to raise awareness about this valley and also to show that it can be defeated. And we wanted to encourage and pay tribute to you, the researchers, and the painstaking work that you do, persevering even when frustration creeps in and results resist to show. We need you. In our case, the challenge helped start ALS research program in our Basque Country, for which we feel proud and filled with hope, because research is the only way to beat ALS and any other disease. So the more research, the better. Awareness is very much needed too, and that's what those of us who are not researchers need to do. From the Death Valley Challenge, a documentary was filmed and we screened it 2 weeks ago in our city of Bilbao. We hope to show it over there once we finish the English version. I want to end by thanking you again and expressing once again that we will never give up. Hi.
All right. Without further delay, let's continue because we have another LS fighter today with us, which is our next speaker, Bernie Zibrich. Bernie is also an ALS patient. He is an accomplished healthcare innovator, strategist, and entrepreneur. He was diagnosed with sporadic ALS in 2020 at age 32. He founded Project Mosaic, a program aiming at bringing a new class of innovative ALS disease models and an AI-based platform for conducting phase 0 technical trials in a dish to market. Bernie and his team are partnering with researchers, industry partners, including iXCells, and ALS nonprofits, including ALS Network, during this initiative. Beyond Project Mosaic, Bernie is also a NEOS certified ALS research ambassador, a member of the ALS Association Board of Trustees, and is a recognized thought leader on ALS scientific strategy. Bernie lives in New York with his wife, Marie Zibrich, and their young son. We are honored to have him and his wife joining us today, and as well as his Project Mosaic team members, Eugene and Tusha. So because of Bernie's condition, Eugene Brandon is going to be present the work and the mission about their initiatives from lab to market, decoding the ALS mosaic to enable precision medicine at scale. Eugene. Oh, really? Oh, sorry. Sure.
Hello. I just want to say that it is such an honor to be here, and and I've so loved hearing all of the other presentations today, and 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 maybe as a way of introducing what we are hoping to do with Project Mosaic. One thing that is incredibly exciting about about ASOs and the precision medicine techniques that we heard about before is that they allow us to target rare diseases at at the source, and and as many of you probably know, and as we will discuss in a moment, one of the biggest challenges with not only ALS but other neurodegenerative disorders like Alzheimer's, Parkinson's, and 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 FTD is that many cases don't have an obvious obvious genetic target to treatment. So a big thing that we are trying to do is develop ways of being able to identify both targets and promising drugs that that might work for subsets of of patients with these diseases. And then the last thing I'd like to say before I hand it off to Eugene is that is that that one thing that I love about working in this field is 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 mission driven and 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 collaborative. So so many people are and and I and I am and I'm so excited that we will also be hearing from 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 Dr. Don Cleveland and 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 Dr. Justin Cheetah because a lot of the of the science that we are now now trying to build on and scale we 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 owe a a big a big a big debt of debt of gratitude to to their research as well.
Thank you. Thank you, Bernie. I want to thank iXCells for inviting Project Mosaic to present today and for having a session on ALS. As Bernie was just pointing out, it's pretty interesting that the rarest diseases, the gene is often known, and then you have to address it in that way. And here we're almost coming in as one of the least rare diseases, meaning there's a prevalence of 30,000 in the U.S., but an incidence that's much higher, equivalent to multiple sclerosis, for example. And in the vast majority of cases, we don't know what the gene is, or if there even was a gene that was involved in the source of the ALS incidence or progression. So here's the team for this is a early stage in this operation, the Project Mosaic, but we're launching fast, as you'll see in the presentation, and want to acknowledge Bernie for all the work he's done to get this to where it is today. I first met Bernie a few years ago when he joined the board of the ALS Association, and he started telling me about this idea of neurobiopsies for patients. And at the time I was a little skeptical that this could be something that would be sufficiently effective and sufficiently cost effective to really attract investigators and investors. But now things have changed. There's the technologies have evolved, and the landscape is different, and there's just things are accelerating. It feels like the whole field of ALS has been accelerating over the last few years, and I think now is the time that a Project Mosaic is actually really needed. So we'll talk about that right now. So because these are Bernie's slides, and I just joined the group, I'm going to read my talk, which I admitted to Don, it's a little bit embarrassing. It's a little unorthodox for scientists to read a talk, but I think that's what I need to do this justice. So what is the vision? What is the motivation? And neurodegenerative diseases are tragic, and everyone is different. Everyone is different. And that's kind of the recurring theme here. What if we could cure devastating diseases like Alzheimer's, Parkinson's, or ALS with treatments tailored to each patient's unique biology, unlocking a future where precision medicine is what transforms lives. So this is obviously a highly ambitious goal, yet we see a path which firstly involves creating an in vitro avatar for each person, which we'll talk about now. We're extremely excited to be embarking on this journey. So where are we today? Let's consider the current state of affairs for neurodegenerative disorders. There are millions of people. This is not rare to have a neurodegenerative disorder. If you consider Alzheimer's with nearly 7 million people in the US, Parkinson's over 1 million, and the other neurodegenerative disorders combined, this is 10 million people in the United States, costing approximately, by some estimate, $650 billion. And this is largely carried by the Medicare, Medicaid, and insurance, but also importantly, families. This is one thing that we come across a lot with the ALS Association, is that families are really taxed financially dealing with their disease. These neurodegenerative diseases, adult onset neurodegenerative diseases, are for the most part sporadic, with no particular genetic cause or even environmental cause identified. So this has been plaguing the field, in particular ALS, for forever. And it's still something that we think we can start to ferret out, as research continues to unfold. Tragically, there are no effective drugs to slow the progression of these diseases for sporadic ALS. So Don will talk a little bit in a minute, I'm sure, about the antisense oligonucleotide for SOD1 ALS. And on top of that, there's a high failure rate. So the rate of drugs not making it to market for neurodegenerative diseases is 10 times greater than for other CNS disorders. So this is really a situation that's screaming for a new approach. So let's consider why drugs are failing. Firstly, there are numerous potential causes of disease, and in many cases, there may be several causes interacting to initiate and propagate the condition. Thus, testing a drug addressing one specific molecular target may be insufficient. Even identifying a target may be difficult. And importantly, the course of disease and the nature of disease can be very different between patients. This heterogeneity is seen in all of the neurodegenerative disorders, but ALS is particularly notorious for it. Typically, it's said that the prognosis is 3 to 5 years with ALS, but from what I have observed with my ALS friends and colleagues, admittedly, this is anecdotal, there seems to be at least as many exceptions to this prognosis, with some pals dying in less than a year from symptom onset and others living a decade or more. Clearly, a one-size-fits-all drug development program is not working for neurodegenerative diseases, and given the heterogeneity, maybe we shouldn't even expect it to work. Despite the heterogeneity with neurodegenerative conditions, research has revealed a common feature across these diseases, and especially prevalent in ALS, which is loss of function of a protein known as TDP-43. TDP-43 is an RNA handling and processing protein, which generally operates in the nucleus of the cell, but in many cases, including over 95% of cases of ALS, TDP-43 protein is found mislocalized to the cytoplasm. And in the cytoplasm, it is prone to aggregation, becoming insoluble and dysfunctional. So as we consider the platform we are building at Project Mosaic, we will pay special attention to TDP-43 loss of function as an important feature. So what is Project Mosaic? Project Mosaic aims to transform the way we view neurodegenerative disorders and revolutionize treatment development, driving more precise and effective therapies. What do we mean by this? Well, let's consider cancer by analogy. 100 years ago, cancer was considered as a single disease which can affect different organs and systems, but which was really best treated with blunt tools such as radiation and surgery, with obviously limited efficacy. Nowadays, we know so much more. For example, within breast cancer alone, there are at least 4 different forms, which can have very different prognoses and susceptibility to treatments. Thus, you will not be surprised that in cancer research, drug screening can include testing on hundreds of different patients' cell lines at a time. The PRISM lab at the Broad Institute is a great example of the kind of initiative and represents an effort which Project Mosaic may seek to emulate. The PRISM lab has over 900 cell lines from different patients and covering different cancer types, with extensive clinical and biological information associated with the lines. Hundreds of collaborators are partnering with PRISM, including over 30 biotech and pharmaceutical companies, reflecting the incredible opportunity to gain critical knowledge using such a powerful system. Project Mosaic aims to do the same for neurodegenerative disease. So why ALS? Obviously, Bernie and I and others have a personal interest in ALS, but at the same time, it represents a great starting point for this approach. That is, a great deal of scientific effort has been invested in ALS, and we already have industry-ready sporadic disease models and commercially ready iPSC cell lines with ex vivo data. As well, ALS pathology is concentrated in known distinct cell types, and the potential for precision medicine to be effective has already been demonstrated with the recently approved ASO to SOD1 known as tofersen or Qalsody. Importantly, recent advances in iPSC-based models suggest that a personalized ex vivo approach is possible. Scientific reports from the Achita lab at USC and the Coyne lab at Johns Hopkins have shown disease phenotypes in vitro, including TDP-43 loss of function and the potential for therapeutic intervention. Yet some barriers to adoption remain. These include demonstration of reproducibility and reliability in the industry setting, questions as to the volume of data that will be needed for regulatory acceptance, and correlations to patient disease characteristics, including responsiveness to approved therapeutics or therapeutic candidates. And reports have shown that, like the heterogeneity of the disease itself, there is heterogeneity in how well ALS patients' cells respond to drugs in vitro. These data suggest that there is an opportunity to stratify patients in other ways than just the stage of their disease or the course of their disease, as we seek to tease the subtypes of ALS apart and improve clinical trials. Project Mosaic seeks to move the approach from academic research to commercial utility. And in November 2024, Project Mosaic had a meeting of the minds to explore the possibilities, consider what issues might need to be addressed, and make a plan to get the project rolling most efficiently and effectively. You can see there was both drug makers, thought leaders, academic labs, government and nonprofit groups, as well as CROs, including iXCells at this meeting. And here you can see Project Mosaic was fortunate enough to attract some of the best in ALS from academics to industry for this meeting in Washington, D.C. last November. So the moonshot outcome of the meeting is a goal to make standardized sporadic ALS disease models and a PRISM-style AI drug testing platform, and to have these available commercially within the next 3 years. We don't consider this just an aspirational goal, we believe that with sufficient resourcing, it can be achieved. So how are we approaching this ambitious goal on a practical level? We need to start somewhere. So we take the crawl, walk, run style ramp up. Learning to crawl in stage 1 involves getting the cells up and running from a relatively small number of donors and establishing the endpoints and the protocols for this foundation of the project. This will culminate when the systems are internally validated and reliable and reproducible with a track record of success. Stage 2 will involve so-called external validation, establishing algorithms to mine the data and assessing the connections between results in vitro and patients' characteristics. Stage 3 will be the scaled clinical trial in a dish, whereby we have a system of sporadic ALS in vitro, which can be used commercially for research on ALS, for drug screening and drug development. Ultimately, we would like to have a system which can be used to predict which course of treatment is going to be most effective for a given patient. We are grateful to iXCells for partnering with us on this. As well, we have fantastic tissue and data partners lined up in Answer ALS, ALS TDI, and Genius, the data company. So we are on track for beginning this work now. And I'm very excited for Neon Way, and we'll be having a meeting tomorrow to keep this rolling. Indeed, we can't afford to wait. The Project Mosaic system is needed as soon as we can get it up and running. And there are clinical trials in ALS today which could benefit from Project Mosaic's inclusion. So, Bernie, let's finally make neurodegenerative disease treatable. Are you in? Thank you.
Thank you, Eugene. Thank you, Bernie. Yes, we are in. Yeah, so I'm very excited to get something done. Next speaker. So we're gonna welcome a prestigious professor, Don Cleveland, who is a distinguished cancer biologist and neurobiologist, renowned for his groundbreaking research in neurodegenerative disease and cancer. And among his long list of titles and awards, he is an elective member of the National Academy of Science, American Academy of Arts and Sciences, National Academy of Medicine, American Academy of Microbiology. Dr. Cleveland is the 2018 Breakthrough Prize recipient in Life Sciences for his pioneering research in neurodegenerative diseases. Don's major contribution includes the studies on tau pathology in diseases like Alzheimer's and other tauopathies. His team made a major contribution in identifying key mechanisms of ALS, as well as optimizing technology for delivering ASOs to the brain and spinal cord, overcoming the challenge of the blood-brain barrier. These findings directly result in several clinical stage ASO drugs developed by Ionis Pharmaceuticals. This includes Spinraza, the first FDA-approved treatment for spinal muscular atrophy, or SMA, as well as tofersen, one of the first targeted therapy for genetic form of ALS, SOD1. So I think Ionis owe you tons of money. So last summer, I had an opportunity to travel with Don and spent a few days with him visiting several ALS patients and laboratories and biotech companies in China. I'm extremely touched by his uncompromising scientific rigor, genuine care and compassion of patients, and deeply committed to mentoring the young scientist. So I've been discussed a lot about whether iPSC model is a really suitable model for modeling ALS. We have a lot of different opinions, which I like. So with the greatest respect, Don.
Yeah, so thank you for that. And what I thought I would do today, I saw the program and I thought for some of the young folks, even the ones at the corporate positions, I wanted the birth of the use of antisense oligonucleotides, or as I call them, designer DNA drugs, in the nervous system. So from beginning to the next ones. Okay, so that's what I hope I'm going to do today. So, and we started in 2003 with the then ISIS, not Ionis, now Ionis, and recognizing that actually for all of the major diseases of the aging nervous system, all the big diseases, Alzheimer's, Parkinson's, ALS, Huntington's, where there was a gene involved known to be causative or at least contributory, they were all widely expressed, some ubiquitously expressed in every cell in the nervous system. And indeed, it was then this two discoveries. The first one by Andy Singleton and his colleagues at NIH, who discovered that if you inherit 3 copies instead of 2, 3 copies of a completely normal gene that encodes alpha-synuclein, you will develop early onset Parkinson's disease. And that was followed by a similar discovery that if you, because of a recombination error, you inherit 3 perfectly normal copies of amyloid precursor protein, a gene encoding that, you will develop early onset Alzheimer's disease. So too much of a good thing. And indeed, and a therapy that would be directly on disease mechanism would be just to, in those instances, would be to lower the level of production because 150% is going to be disease causative. So we set out how can we do that? And of course, for this audience, probably everybody realizes, well, ASOs are going to be a way to do it. But if you have, and I apologize to you, an even younger audience, and they're young scientists, they're going to tell you use an siRNA or an shRNA, and you'll tell them, no, we're going to use something older. Older, the idea that if you can make a single-stranded piece of DNA, easy to make, and then if you can get it inside the target cell so that it can pair with a target RNA inside the nucleus of that target cell to make an RNA-DNA hybrid, we can then exploit the presence in almost all of our cells of a nuclear enzyme, RNase H, which cleaves the RNA, but not the DNA in this hybrid. And therefore, it's a catalytic RNA destruction mechanism that relies upon a single endogenous enzyme that is present in almost all of our cells anyway. And over the years, the medicinal chemists, we heard a discussion of that earlier this afternoon, have modified both the DNA backbone and the 2' and 5' positions of the ribose sugar to produce the kinds of ASOs that are now being used in human trial. But it was actually a very sobering problem, was that if you dose in the periphery, by any mechanism that we know today, effectively 0% gets in the nervous system. So what we're going to do is to deliver it into the cerebral spinal fluid near the bottom of the spinal cord. And that's what you heard it this afternoon, intrathecal administration. And we did that after we had failed to use pumps for continuous flow. We went to Medtronic, and their conditions for using their pumps were so unreasonable that we actually thought the whole project was dead. And then I got my big team together, 3 of us. You'll see them in a minute. The 3 of us, and one of us said, well, why don't we just inject it in and see how long it lasts? Because we should have done that right from the beginning, but we hadn't. And that's when we learned that these modified DNAs, that you can, the best of them, that you can get from single dose injections, you can get three to 4 months worth of efficacy, and that we didn't need pumps after all, that we would just use periodic dosing. Okay, so, and we discovered here in that if you do that intrathecal dosing, and this is work from, first part of it was from my team at UCSD, and now it's Holly Cortesewicz, who is the head of neuroscience at Ionis now, and a second publication from Ionis. Yes, if you dose intrathecally, you can hit almost every region of the non-human primate brain effectively, near the site of injection, but you can see all the way across, although you miss some very deep structures. So you can't get everywhere, but you can get almost everywhere. Okay, so, and the disease, yes, that we started with was ALS. We, of course, you've just heard about that. And the founding example here in this paper from 1993, now, wow, 30 years ago, the original proposal, there were mutations in a gene called superoxide dismutase, and those mutations were proposed right here in the News and Views article accompanying the original publication. They were going to be loss of function because mutations usually lead to inacting or partially inactivating the gene. And that's what, and we tested that. We made, of the original 11 mutations, we made 6 and tested them. And one of them, this one, with this amino acid modification, has 0 activity as a dismutase. And then we asked, does it cause disease in mice that have 2 endogenous genes? They have normal dismutase activity. And the answer is they develop age-dependent fatal motor neuron disease. And then we removed one of the endogenous genes. Oh, and then we removed both of the endogenous genes. So we have mice with normal dismutase activity, half 0 in the presence of an inactive mutant, and it doesn't change the disease course at all. And that led, after a contentious scientific time, that led to what is now a consensus view that these mutations provoke disease through acquisition of one or more toxic properties, not through loss of enzymatic activity, although it actually is quite sobering that in 2025, we do not agree on what the key toxicity is. But if you could just turn the gene down, you'd be directly on disease mechanism. And so we set out to do that. And this is the founding use of trying to use an antisense oligonucleotide in a therapeutic way, taking a transgenic rat that expresses an ALS-causing mutation in superoxide dismutase, and now dosing it, and here, near the site of injection into the cerebrospinal fluid, yes, you can, in a dose and time-dependent manner, lower the level of the target RNA from blue to yellow. And oh, look, throughout the entirety of the the rat nervous system. And if you dose these animals, it's a hyper-aggressive model of ALS-like disease from mutation in SOD1, but here's the founding experiment. And if it hadn't been successful, there wouldn't have been a second experiment. But this is it. And we took animals that were either mock dosed or dosed with a saline, and we dosed them at disease onset. And I said it was hyper-aggressive model. They only live about 25 days before you have to euthanize them for humane care. But look, we doubled survival after onset in this animal by dosing at onset. So that, and we thought, hey, we've actually done something. We've really been able to target the actual cause of the gene. And indeed, just in 2023, got FDA approval. Here was the trial. You can see loss of muscle strength in the green curve are the dosed patients. The dashed blue curve is were the placebo patients. And now you can see, oh, look, in the open label extension, 6 months, the original trial was for 6 months, but in 6 months afterward, 25% of the patients got stronger. They got stronger. This was a demonstration that ALS is a treatable disease. And even the placebo patients, when they were converted to drug in the open label extension, you can see they are also following the flattening of disease course. And we now need to follow these even more carefully, but there are anecdotal examples of individuals with mutations that really cause very rapidly advancing disease who have lived much longer than had been recorded by any of the clinicians I know, any of the neurologists I know. So and indeed, 2023 was a watershed year, in my view, for ALS, because there's evidence for SOD1 mediated ALS, and even in a second inherited cause of ALS, mutations in a gene called FUS. That work led by Neil Schneider at Columbia with a series of colleagues, including Sandrine de Cruz, who was here in San Diego at the time. Okay, so now I'm just going to take a further view of what's happened in the ASO space in neurodegenerative disease, and then come back to how can we use that in ALS, okay? So the most frequent genetic cause of the second most frequent disease of the aging nervous system, Parkinson's disease, is mutation in a gene called LRRK2. And LRRK2 encodes a kinase, an enzyme, but these mutations are not inactivating mutations, they actually hyperactivate the kinase. So if you could just turn it, and if you can just turn the gene down, you would be directly on mechanism. And with Ionis and Biogen, that has now gone to trial. And here, thank you for the introduction. Yes, I did get the Breakthrough Prize. It was handed to me by Sergey Brin, the founder of Google, who, this is very public knowledge, so there's no HIPAA violation here, who is the most famous LRRK2 mutation carrier. His mother is a homozygote. And actually, I missed a huge moment. I did not know he had LRRK2 mutation at the time I met him, but we have gone to trial for his mutation. And indeed, I opened with the idea that if you inherit 3 copies of a completely normal gene encoding alpha-synuclein, you will develop Parkinson's disease. And alpha-synuclein misaccumulation is found almost universally in sporadic Parkinson's patients. And so the idea of turning down using an antisense oligonucleotide to turn down alpha-synuclein, that too has gone to trial with Biogen and Ionis. And I can't wait for the outcome. So I have no affiliation with Biogen, and I have been a consultant for Ionis since 2003-ish. So I should have pointed that out on the first. I own not one share of Ionis stock. Okay, so, and then there was one other disease, the biggest disease, Alzheimer's disease. And this is the 46th anniversary of the discovery of a protein called tau. You can see there was this young graduate student and the tau misaccumulates inside the affected neurons in Alzheimer's disease. And actually, it's the best predictor today of cognitive decline. So you probably are aware that there are 2 major pathologic events in Alzheimer's disease. There's accumulation of a piece of one protein, A beta, accumulating outside the neurons, and there's tau tangles accumulating inside the neurons. And you know, it's like if there's a garbage strike and the garbage is piling up outside your house, that's bad. But if there's a sewage problem and it's backing up inside the house, don't you think that's worse? And and indeed, it went to trial to try to suppress tau. Let's just try to suppress tau. And you can see this is 2023. And indeed, with these ASO drugs, you can you can lower the level in a dose and time-dependent manner, lower the level of total tau or phospho-tau for those who think that's the more toxic form. And indeed, but the most exciting part is here. There is imaging, positron emission tomography, PET imaging that can identify the tau deposits, at least some of the tau deposits. And look, here's 2 individuals who were entered into the trial in the placebo arm. And what happened is that, oh, look, they have these deposits, the reddish yellow stuff, these deposits. And the deposits got bigger in the first 6 months of placebo treatment. In both cases, they got bigger. And then COVID disrupted much of the imaging, but then they were imaged 1 year later after converting, being placed in the drug arm to suppress the synthesis of tau. And look what happened. So the deposits almost disappeared. So that was true for 11 of 12 patients. Again, it's not the imaging was really disrupted by COVID, but the idea 11 out of 12, it's actually really quite good. I could hardly have drawn it better. But Biogen released no information about cognition, but they have pledged to do so this year. So I can't tell you. I don't know either. But anyway, it will be. And now, okay, so we now know we can do this. What about ALS and sporadic ALS? And you heard it before that there's this protein that's typically intranuclear, the brown stuff here inside this nucleus, but it accumulates. Oh, look, it cleared out of the nucleus here and accumulated in this big deposit over here in that cell and another one over here. And so this is the hallmark of actually multiple neurodegenerative diseases, but it's absolutely the hallmark of sporadic ALS. And also the second most frequent the most frequent genetic cause of ALS. That has these TDP43 abnormalities. Now, so we tested, well, what happens if you lose TDP43 from nuclei? And so we suppressed it. We suppressed TDP43 to about 25% of its original level, and we saw that there was an abundant protein that was suppressed even more, and it's a protein called Stathmin-2. We know this is actually an abundant RNA in human motor neurons, and it had been proposed to be required for recovery after axonal injury. And I'm going to show you, it's absolutely required for recovery after axonal injury. So, and we know why it gets lost, but when TDP43 binds to its pre-mRNA and blocks the use of a cryptic splice site. And so that if you lower TDP43 here to about 25% of its original level, Stathmin just disappears as a protein, completely disappears. And we know it's because it gets cryptically spliced and polyadenylated. And indeed, if you ask what happens if you look at the Stathmin RNAs, here you go, exon 5, exon 4, exon 3, exon 2, in a typical RNA-seq experiment. Yeah, there they are. But if you suppress TDP43, look what happens. You miss the protein coding exons, and you now use this cryptic splice site, which truncates the RNA, making a completely non-functional RNA. So we know that works, and even more so, if you go to sporadic ALS patients and recover the surviving motor neurons from an autopsy samples, you recover them and ask what happens. Well, if you take individuals who died from non-neurologic disease and you look for Stathmin, you'll see exon 5, exon 4, exon 3, just what you saw before in cell culture, but look in the sporadic ALS patients, they're all truncated. They're almost completely truncated because of loss of function of TDP43, and that was true in 13 out of 13 of the sporadic ALS patients that were tested. Okay, so, and even more so, we have identified in 2023 that binding of TDP43 at that locus in the Stathmin pre-mRNA is absolutely, that TDP43 acts just to inhibit the use, the 3' splice site finding machinery from identifying that cryptic site. Okay, so does the loss of Stathmin, is it really a key component of driving ALS-like disease? And so what we did, was Yoni Lopez-Raskin right there. What she did was to suppress Stathmin in completely normal mice, really aging normal mice. And when she did that transiently using an antisense oligonucleotide, look what happened. The neuromuscular junctions where the motor neuron and its axon connects to the muscle. And most of us think that in ALS, that's the disconnection is the first step. And here, oh yeah, you drive disconnection by transient suppression of Stathmin. And if you suppress it even longer here for 8 months, oh yeah, you have more than half of the neuromuscular junctions are now disconnected. So loss of Stathmin is sufficient to cause the first steps of ALS-like disease. So, okay, so we argued that. And now you heard about this again earlier this afternoon, and I put this slide back in just to remind everybody. In in spinal muscular atrophy, it's a disease. It's a disease completely of the motor neurons. It affects, and the affected kids have inherited 2 inactivated copies of this gene, the SMN1 gene, but we, almost alone among primates, have a second gene. And it encodes exactly the perfect protein. Oh, but its RNA gets misspliced to make a non-functional RNA. And therefore, these kids have both genes are basically inactive. And now, so the strategy was now, can we use an antisense oligonucleotide, which is modified so when paired with an RNA is not recognized by RNase H. And so it's not cleaved, it's simply blocking actually encouraging the use of the correct splice site. And here, if you take cells from one of these individuals, the RNA is almost completely missing as an essential exon. But if you use this in ASO, you can now restore it almost fully. That's the basis for the Breakthrough Prize in 2019 to Adrian Craner and Frank Bennett. Adrian Craner at Cold Spring Harbor and Frank Bennett, the chief scientific officer of Ionis, because for the first approval of an ASO in the human nervous system, a true disease-modifying therapy. Okay, and we're going to try to use that strategy in sporadic ALS. And what we're going to do is to make cells in which carrying, actually this is an ALS causing mutation in TDP43. We incorporate it into both copies of the TDP43 genes in human cells, and then ask here, by tiling with Ionis across this region, can we find ASOs which will block the use of the cryptic splice site when TDP43 is absent? And the answer is, well, you first ask, can you suppress the use of the cryptic splice site? Yep, you can suppress it in a dose-dependent manner, as you'd expect, with both the blue ASO and the red ASO. You can see they're positioned here. And even more importantly, they restore the correct level of the Stathman encoding RNAs here, starting from 50% of the normal level, bringing it back almost to 100% with both the blue or red ASO. Okay, so we think we have a drug that can restore Stathman even in the absence of TDP43. Is that functionally important? And so to do that, what Yoni and her colleagues did was to grow human motor neurons in these compartmented chambers where there's cell bodies are over here. They have their axons and processes into the distal compartment, and now you can intentionally damage them. You can axotomize them, cut the axons off mechanically, and now ask, can they recover? And the answer is, yeah, they can recover. If they're normal human motor neurons, they just recover beautifully within 24 hours. They regrow those damaged axons almost perfectly. Now, can you and here's another example of that using the control ASO. Yes, after axotomy, they recover. Look, the processes recover, looking at either the presence of Stathman or a neurofilament. Now, if you now suppress TDP43, and ask, what happens? Can the axons recover? And the answer is, you can see, no. Actually, the neurons are still there, but they don't recover at all, at least not in the short term. Now the question is, can we use a rescuing ASO to restore the synthesis of Stathman, even though we're not restoring the other products, the other RNAs that are misprocessed when you lose TDP43? So we're only putting back one of the misprocessed products, and it almost completely restores it. So we argue that we have proof of principle here in human neurons in culture that restoring Stathman, even in the continued absence of TDP43, can really make a benefit. Now, one last thing. So it was my group and Kevin Egan's group then at Harvard who in back-to-back papers identified Stathman as a target in ALS since 2019. We achieved last year, we humanized the Stathman allele in mice so that we could test, do we have an ASO that can restore in the mouse nervous system, can restore Stathman synthesis even when TDP43 is not bound to the Stathman pre-mRNA, and we identified that in 2023. We hope with Ionis, we hope to get to clinical trial this year. and I should mention that a small startup, Curalis, went to clinical trial with an ASO already 2 years ago in 2023, and we're I want to make it really clear. I wish them the best. I hope their ASO really does in the actual nervous system restore Stathman, and that nothing would make any of us, I bet anybody of us in the room, happier than to see a true effective therapy by anybody. Of course, we're even happier if it's us. But okay, so all right, so here's the scorecard of where we are. We know we can use these ASOs in broad and deliver them broadly throughout the nervous system of rodents, non-human primates. We can use them for catalytic mRNA degradation or altered splicing. The best of them have very long efficacies. We know they're certainly commercially feasible, and they can be safe. You have to screen for the safe ones, but you can be. The first approval was in spinal muscular atrophy, 2016. Second approval from SOD1 mutant-mediated ALS in 2023. There are ongoing trials as I indicated, in Alzheimer's disease, lower tau, 2 in Parkinson's disease, one of which we'll hear about. There'll be a readout from the trial later this year. There were also 2 failures, and there were 2 failures in ALS. There was a target. The most frequent genetic cause is a hexanucleotide expansion in the gene C9orf72, and that failed despite lowering C9orf72 as expected. No clinical benefit, and the team, there's an intra-team disagreement about what is the source of the failure. All right, so there was another one in Huntington's disease, 100% genetic disease from dominant inheritance in the same gene. That too lowered mutant Huntington, absolutely, and absolutely had no clinical benefit. In fact, possibly, and again, the overall collective effort, we do not agree on what was the source of the failure. Roche ran a trial, a 700-patient trial. They're running a second 350-patient trial, now using even a lower dose of that ASO and dosing only patients that were much earlier in disease. They're fearing that they just started too late, lowering the power to show at real efficacy. Okay, so that's where we are today. And I just have, yes, okay, so who was the we that I described today? And this is the founding quintet of the use of an ASO in the human nervous system. This local neurologist, Richard Smith, was absolutely adamant. Some of you have met him, you know that he's absolutely adamant and he without this, it would not have started. Tim Miller, who was a postdoc with me at the time, co-ran the clinical trial for SOD1. My animal coordinator who did the surgeries to introduce these ASOs, and Frank Bennett, the chief scientific officer of Ionis, and the effort to identify Stathman as a target. A pair of postdoc fellows with me, Yoni Lopez Oraskin, who's here, Zeev Melibin, now on faculty in Israel, and a pair of former graduate students now graduated. And my long-term collaborator, Clotilde Lagier, and her team at MGH. Okay, so thanks very much.
Thank you, Don. It's always inspiring to hear your. So actually, I learned quite a bit about the TDP-43. The proteinopathy when I talked to Project Mosaic team, that's my cheat sheet from their team. So I really learned quite a bit. So our next speaker, so we will have our next speaker, Justin Ichida. He is a prominent stem cell biologist and neuroscientist dedicated to advancing treatment for neurodegenerative diseases. He's an associate professor of stem cell biology and regenerative medicine at the University of Southern California. In 2022, Dr. Ichida joined BioMarin Pharmaceutical as a VP and head of neuroscience research. He's also a co-founder of Stem, a biotechnology company focused on developing therapies for neurodegenerative diseases. His research primarily involves creating in vitro models of human neurodegenerative diseases by converting patients' skin fibroblasts into disease-affected neuron cells, facilitating the discovery of potential therapies. Without further ado, Justin.
Thanks. No, I think my conclusion from that is I have too many jobs, but it's really a pleasure to be here in what feels like the epicenter of ASO technology in the CNS, and I've certainly drank the Kool-Aid for this modality as well. So I'm going to talk about some, as you mentioned, I am the head of neuroscience research at BioMarin, whose mission really is to develop therapies for rare disease. Today, I'm going to talk about some research from our lab at USC, much along the lines of the Project Mosaic talk earlier, in seeing how we could use iPSCs and leverage them to maybe identify points of convergence in sporadic ALS, and also to better leverage the genetic information that might be out there to point us in the right direction. So this was a perfect setup the last several talks, but we definitely think of ALS as a landscape with many different etiologies of the diseases. We do have, some known genetic forms. They might be clustered around mutations that cause instability or misfolding in proteins like TAR-DBP or TDP43 that are already prone to misfold. On the other hand, you might have mutations in genes that affect the protein clearance mechanisms within neurons, which could also lead to aggregation of things like TDP43. And then even some cytoskeletal genes, which may encode proteins that underlie the protein clearance machinery and their ability to function. And I think it's a perfectly good and sound strategy, and maybe the best strategy, to go after these known causes of the disease, maybe with designer ASOs. I think there are many people, or some people, going after that, and I think that has a high chance of success in some cases. One of the challenges is that we have this unknown or sporadic sector of patients where, as you can see, it's the bulk of cases, and we don't yet know all of the genetic causes that might underlie those diseases. In fact, we don't even know that there are genetic causes of those diseases. And therefore, it's been really difficult to try to understand if things that we're developing for some of these rarer forms or testing on models of the rare forms really would translate to a mixed population like you would see in a sporadic ALS clinical trial. And this is what we're trying to potentially use iPSCs for, is to understand what therapeutic targets might be out there. Are there any therapeutic targets out there that might target a point of convergence with all these forms of ALS? As I mentioned, the genetics is not well understood for 90% of ALS patients, but we know that we can make iPSCs from these patients. And if there are genetic drivers within those patients' DNA, they will be preserved in the iPSCs and the motor neurons we make from them. And if we turn them into motor neurons, they may drive disease. I think that was like the overall hypothesis. And if so, we might be able to learn about, what are the differences in different forms of ALS, but also what proteins or pathways could we target to reverse some of the pathology that affects all patients. What we did to start, it was pretty unknown, of course, that iPSCs or motor neurons in a dish that are maybe a month old would ever be able to mimic what happens in a patient's motor neurons when they get ALS in midlife. But we nevertheless tried it, and this is a big graph bar chart here, just showing the rate of death of neurons made from iPSCs from these different individuals. So on the left, the far left, there are about 9 white bars. Those are all motor neurons made from unaffected control. individuals, and they had a hazard rate or rate of death that we sort of averaged out to be 1. And then the next set of bars, the brown bars, that's the hazard rate, the rate of death of neurons made from C9orf72 ALS FTD patients. And you can see they're uniformly higher than the average of the controls, which goes along the lines of their genetic lesion in C9orf72 potentially driving their faster motor neuron death. And then the next set of bars are motor neuron survival times from different sporadic or unknown ALS cases. And I think what was very surprising to us was that all of the lines that we tested showed a faster rate of death compared to controls. And then on the far right, there are more sort of rare genetic forms of ALS, including mutations in TDP43, also having the same sort of phenotype in a dish. And so you could also see on the left that if you look at the TDP43 within those neurons, in the control samples, they're all predominantly nuclear. In the C9 and sporadic ALS cases, you see a lot of that TDP43 leaking out and being sort of deposited in the cytoplasm and leaving the nucleus. So this is essentially what one sees to some extent in postmortem tissue of ALS patients. Even though we didn't know that there would be genetic causes of any of the sporadic ALS cases, I think one of the simplest interpretations of this data is that there are likely genetic drivers of motor neuron disease in many of these, in all of the ones that we tested. Maybe it's polygenic, and so it's been difficult to really pin down all of the variants that contribute. But this suggests that we may be able to use these lines to understand how motor neurons from different types of ALS patients could react to the same type of therapeutic perturbation. Not to beat a dead horse, I'm definitely preaching to the choir here, but one of the convergence points we may be able to target would be the TDP43 pathology. We like that as a target because TDP43, the gene encoding TDP43 can harbor mutations that have been shown to cause ALS, and this pathology forms in 95 to 98% of patients, but it's not, as Don eloquently introduced, it's not that simple to target the pathology because you don't want to knock down the gene because it has an important function in splicing. And if you try to kind of overexpress it to kind of restore nuclear function, what's been seen in the literature is that slight overexpression can actually lead to more pathology. You sort of get the aggregation that you see here in the cytoplasm. So it's a tough nut to crack, I would say. But I think it's something that we're game to try. And so how do we sort of go after things that might address TDP43 pathology? It might act upstream of it. We decided to do a screen, a genome-wide screen, using lines from a TDP43 patient. So this is an ALS FTD family where they harbor a single mutation in TDP43. As you can see by this schematic, I think one of the theories in the field is that many of the mutations that cause ALS in this TDP43 protein occur in this prion-like domain that causes it to aggregate more than it normally does. And that could lead to the pathology that one sees. But even in cases where there aren't mutations in that gene, it's thought that things that lead to higher concentrations of TDP43, like deficiencies in protein clearance or other types of stress, could also lead to this type of aggregation. And we thought we could model this potentially in the iPSC neurons as I showed you previously. And so Jasper, Ruben Sigler, and Sharon Tu in our lab started collaborating with Ken Kosick and Juliana Acosta Uribe, who had in Colombia found what I think is one of the largest TDP43 ALS FTD families known to date. So they have a spectrum of different ALS FTD phenotypes, but they all have the same mutation in TDP43. and we made iPSCs from that patient line. And what we did was we generated these spheroids of neurons from that patient or from controls. As you can see on the left, if we look at like the known genetic form of ALS, C9orf72 patient neurons, they have a fast loss of their motor neurons in a dish compared to controls. And similarly, the neurons from this TDP43 ALS FTD patient have also exhibited fast decline. So this was kind of a phenotype that we thought we could search for genes on where, if we, with an eye on maybe ASO, antisense therapies in the future, could we find genes that, when suppressed, could rescue this type of degeneration. And so Jasper set up a genome-wide screen using CRISPR technology. And so I think we had done chemical screens like this, 6 or 7 years ago. It probably took us, 5 months to screen 2,000 compounds and pathways. This one took us like 2 weeks to screen all of the genes in the genome. And so essentially what we were looking for was, every neuron got a different guide RNA that suppressed one gene. And then we were just looking for the surviving neurons at the end of the survival assay and to see which guide RNAs were most enriched in those. And that would suggest that the suppression of those genes promoted the survival of these neurons. So we found a lot of things that we had seen before, like PIC5. We had seen things that other people have found in the literature, like nuclear porin genes. We also found a gene called VAV2, which caught our eye, because what we wanted to do was really triangulate this with human genetic data to see what things might really modify disease in humans. And so Michael Snyder's group had just recently published a paper where they essentially leveraged iPSC motor neurons and said, motor neurons are likely the causal cell that's affected in ALS. And so what we should do is really only concentrate, or one approach is really to concentrate on the motor neuron genes and the regulatory regions that regulate the motor neuron genes, and sort of eliminate all of the other variants that are just contributing noise or decreasing our power of genetic analysis in this already sort of rare disease. And when they did that, they found genes that were really enriched, either in the gene themselves or in the enhancer region of the motor neuron gene, enriched in genes that were differential, enriched in SNPs that were differential between people that had ALS and controls. And the top gene that they found where there were either lower VAV2 expression or loss of function variants in the gene that was enriched in controls was VAV2. So what that meant was that regulatory regions of VAV2 were enriched in variants that lowered the gene in controls, and in the gene itself, there were loss of function mutations that were more often in controls than ALS patients. On the right is some data that they pulled from this Answer ALS collaboration, where they made 1,000 iPSC lines from patients, ALS patients, and then made motor neurons from them. What was interesting is that in the motor neurons from that study, they found that the motor neurons from patients where they had lower VAV2 expression had a later age of onset, tended to have a later age of onset. So this suggests that whatever is in the genome, if it causes lower VAV2 expression, they might have later age of ALS onset. So less VAV2 is good to protect against ALS, essentially. We also looked in some existing clinical data from Eddie Lee's group, who had essentially taken brains of FTD or FTD-ALS patients at autopsy and sorted out the nuclei from the neurons and then stained them for TDP-43. And so they could actually do the RNA-seq on nuclei that from neurons that either had TDP-43 or didn't. And what we found was that in neurons that didn't have TDP-43, they had much lower VAV2 expression, they tended to, than the TDP-43 positive neurons. So this could be because TDP-43 regulates VAV2, but in another published data set from Kevin Egan's group, when they knocked down TDP-43 in iPSC neurons, they did not see a lowering of VAV2. So it's not regulated by TDP-43. And so one interpretation of this is just that the TDP-43 negative, loss of function neurons that survived the latest actually had lower VAV2 levels in the brain. So as I mentioned, we're really interested in this ASO approach for many reasons. I think it's a very rapid way to test a therapeutic target in patients. And there are some other benefits, like, you might not knock down VAV2 too much in peripheral tissues compared to a small molecule approach. So as a tool and as a test case, we started testing this ASO approach in vitro. So we made ASOs that suppress VAV2 by around 50%. And then we tested them to see if they could rescue the survival of C9 or 72 ALS motor neurons, about 6 different sporadic ALS lines, and the TDP-43 patient line that we started with. And in all cases, treatment with the ASO dramatically improved the survival of these ALS patient neurons across these different forms of ALS. VAV2, we're still trying to find out exactly how this works, but it turns out that VAV2 can activate multiple sort of GTPases downstream of it. Some of the things, interestingly, that regulates is protein clearance, protein acetylation. And so there may be multiple ways in which this single target is activating protective pathways. We do know already from the graph on the right that each of these different downstream targets of VAV2, when they're suppressed, as you would happen when VAV2 is suppressed, they all can contribute to improved neuron survival. So again, it seems like a single target acting through a multi-pronged downstream protective mechanism. What happens when we look at TDP-43? I think one of the things we're interested in is seeing if we are upstream of TDP-43 pathology and remedy that, because there are dozens of genes like STAthmin2 that when TDP-43 is lost from the nucleus also are lost as a result. And so what you'd really like to do is restore proper splicing for all of those targets. And so what you can see here in the case of the C9 ORF72 motor neuron, as we saw before, there's a lot of TDP-43 out there in the cytoplasm, but treatment with the VAV2 ASO seems to restore that protein to the nucleus, which is really what you want. We're beginning to now look at STAthmin2, for example. As Don introduced in the last talk, there's a cryptic splicing event that occurs in the absence of TDP-43 from the nucleus. And with suppression of VAV2, we can suppress that truncated STAthmin2, that cryptic exon event, and begin to restore STAthmin2 levels. And I think we needed some optimization to look further at that. But I think we're seeing trends in the right direction. We've gone on to see how this might work in an in vivo model. We made an ASO for the mouse that suppresses VAV2 by about 50%. And this is a mouse that overexpresses human TDP-43, so very rapidly develops the loss of nuclear TDP-43 and the cytoplasmic inclusions that you see in patients. They also start having gait impairment very rapidly. So you can see the black line is the increase in gait impairment in these TDP43 mice without the ASO treatment, and in blue is the treatment of the VAV2 ASO. You can see that's quite a bit lower, and the survival time, on average, they improve by about 50% survival, but some of the animals have lived much, much longer. And we're looking into that. We think they have better biodistribution than some of the other animals in the study for the ASO. So that, and then what we also see is that the number of motor neurons is preserved in the spinal cord when we suppress VAV2 in these mice. We also see that neurofilament from the serum starts reducing even in this early time point of the study. This is, of course, a really important biomarker that looks at axonal degeneration or neurodegeneration that we think will be important clinically. So that's one example of using the iPSCs. And then in just a couple of slides, I wanted to illustrate a very similar but slightly different way in which we're using the iPSC data. So in collaboration with Sai Zhang and John Cooper-Knock, they have been looking at the genetics and these rare variants and how they relate to survival of ALS patients instead of likelihood of getting the disease. What's interesting is they found a variant next to a gene called CCDC146, and they predict from the enhancer map of motor neurons that this regulates CCDC146. But the interesting thing is that patients that have this one variant basically survive like that blue line, whereas the average survival of ALS patients is the red line. So they essentially, they go downhill extremely quickly. And CCDC146 is another hit that came up in our screen, so we looked at that further. What it turns out is that that variant actually seems to regulate CCDC146 expression in motor neurons. And so if you look here, the variant that is in the patients that decline very quickly leads to a slight increase in the expression of CCDC146. It also leads to shorter neurites in the neurons made from those patients. If they correct that variant to the other variant, they get much longer neurites. And also this variant that's in the patients that decline quickly, that leads to higher CCDC146 expression. It leads to higher TDP43 aggregation in these cells. And so that suggests that potentially, increasing a variant that increases CCDC146 levels causes very rapid decline for ALS patients. What happens if you now suppress CCDC146? So we made motor neurons from patients, C9 patients and sporadic patients, and suppressed CCDC146 by 50% with an ASO. And that now rescues the survival of those lines similarly to the controls. And then if we use that same mouse model and suppress CCD146 in those mice, you can see that they live quite a bit longer than the negative control treated mice. So, in conclusion, I think that there is a lot of potential for using, familial and sporadic ALS induced neurons to mimic some of the key hallmarks of ALS. I think it's probably not mimicking what happens with, aging of protein clearance mechanisms in humans, but very good at maybe understanding some of the, root causes of pathology in ALS patients. And using a genome-wide screen and convergence with genetics, we found that these two targets might be highly neuroprotective, at least they are in model systems. And so we're looking forward to seeing if we could advance ASOs against those targets. A lot of this work, most of this work was done by Jasper Rubin Sigler and Sharon Tu in our labs, two great scientists. Some of the legacy people have also contributed to this. And our collaborators, Sai Zhang and John Cooper-Knock, have been working together nicely with us on the CCDC146 story. So thank you very much.
Thank you, Justin. We've been hearing a lot of scientific reports. Now we're gonna switch gear a little bit Our next speaker, Sherry Straw. She is the president and CEO of the ALS Network, formerly ALS Golden West, one of the largest nonprofits in the nation focused on ALS. With a master's degree in public health and another in business administration, Ms. Straw has excelled as a nonprofit leader, community advocate, and public speaker. Ms. Straw has been on the executive teams of several of the largest nonprofits in California, providing critical strategic visioning and effective leadership. Ms. Straw currently serves on the board of two highly respected nonprofits, ALS United and Child and Teen Counseling. I met Sherry the first time at a reception of the Death Valley Challenge. I was impressed immediately by her distinctive and charismatic leadership. She's great at making people cry and laugh. So let's welcome Sherry.
Thank you so much. It's so great to be here with all of you. Thank you to iXCells for having us. And what a day it has been. I've learned so much from all of you. So thank you for that. It's very generous. And thank you for setting that up. So, okay, I need more hands. So here we are. I'm sharing about the ALS Network today. I'm going to tell you a little bit about who we are, what we do, giving you an idea of the patient advocacy part of the ALS ecosystem. And then I'm also going to tell you a little bit about how we interact with industry and what our research program looks like. And you can read our mission right there for you. I'm not going to read it out loud. You've got that. And I do want to point out that ALS Network, it's a newer name in the ALS nonprofit landscape. And you may, if you've been around ALS for a while, especially in California or Hawaii, you probably know us as ALS Golden West chapter of the ALS Association. We did leave the ALS Association a little bit more than a year ago. And with that, we changed our name to ALS Network. Fret not, we are the same organization that has been proudly serving the ALS community for more than 30 years. We are deeply, deeply mission driven. And we are passionate about the work that we do. You will all relate very much to this. Lives are at stake and people are counting on all of us. So we work with a sense of urgency. We tend to be very driven, very nimble. We're not afraid of change. We're not afraid to do something in a completely different way. We're known for being innovative and also very collaborative. People often share that, it's so easy to work with you and we love working with you and thank you for collaborating with us. And we usually feel the same exact way. And, the ALS community comes first in everything that we do. And that is the perspective from which we frame our decisions. We work through 3 mission pillars. I'm going to tell you a little bit about all of them. One is care, one is advocacy, and one is research. But first, let me tell you a little bit more about our scope and scale, if you will. You heard Nyanwe mention that we are one of the largest ALS nonprofits in the nation. We rank around 4th if you remove the nonprofit hospitals and medical centers. We do directly serve more than 2,000 people with ALS each year. We also serve the entire family ecosystem. So if you take that number of people with ALS that we're honored to work with, we're also working with loved ones, caregivers, family members, and oftentimes friends. There are people who are completely alone with ALS until we meet them. We make sure no one is alone with this disease, and we mean it when we say that. To date, we've raised more than $19 million that's been invested in ALS research. More than 50,000 different advocacy actions have been taken nationwide. And we do have 3 physical locations. 2 are in California. 1 is in Hawaii. But we really work across the region with staff working remotely throughout the state and across Hawaii as well. So you're gathering California and Hawaii. Yes, that is where our in-person boots on the ground services are focused. However, because we serve so many people with ALS, we do have programming that's relevant for others who live in other places. These are things where we have the critical mass and other regions don't. So anybody from anywhere can come and participate in those services. And an example, a couple of quick examples to kind of give you, what does that really mean? We have support groups for people under 50 with ALS, unpartnered people with ALS, LGBTQIA plus people with ALS, Spanish language support groups for people with ALS, and so on and so forth. Anyone from anywhere can come on in and find community with us. And as I mentioned, we're known for collaborating across the ALS ecosystem. And, we are the ALS network, but there are other ultra super duper, I'm creating a new ultra super duper rare diseases that mimic ALS. These are people who often don't find themselves in any of the other organizations. So we do also serve people with diseases that mimic ALS. So moving right along, let's start by talking about our care services and what do I mean by that? What is that all about? And again, tying back to, I think it was Yael's presentation earlier about patient advocacy organizations and the power of what they're up to. And the beauty too of what your patient advocacy organizations are doing is we're helping our medical professionals, our research professionals focus on their roles, and we can step in and bridge those gaps so everyone can keep their eyes on the prize, right? So every person with ALS that we're connected with is provided with a regional care manager. And these are people in their communities who know where's a good place to go where I can come in with a power wheelchair and get a haircut. I mean, really simple things, and also very complex things like navigating ALS, which is different, as you've heard, different for everyone. So these care managers provide concierge one-to-one services for people and families throughout their entire ALS journey. And this includes doing things like safety checks in the home. And those safety checks are important, again, throughout the journey as ALS progresses, providing referrals, providing information, social and emotional support, help navigating benefits. This is a very complex medical system that we live with here. And there's a lot of assistance that's needed for people to really maximize those benefits. We know that the out-of-pocket costs for someone with ALS can go as high as $250,000 a year. So really leaning in and maximizing benefits is critically important from early on in the diagnosis. I mentioned those support groups. We host more than 30 each month. And we also have equipment loan programs, and these are really important. This can be very expensive, durable medical equipment and communication devices, either things that are not covered by insurance at all, which is all too common, or also all too common, lengthy delays once the prescription has been written for the piece of equipment. We help loan equipment so people can start accessing it right away, even if they aren't able to get it right away through their providers. We are also proud to host educational webinars through what we call our Ask Me series. They live in a library online, so people who are newly diagnosed can access a webinar from years back that's relevant to them today. So a lot of educational materials and resources. And if you know someone with ALS, or you find yourself interacting with someone with ALS, feel free to connect them to us at the ALS Network. And if they're not in a place where we provide support, we will happily connect them with their local patient advocacy group too. Okay, so advocacy. Oh, my friends, is there ever a need for advocacy? Today, in fact, today is a day you can do. I'm going to invite you to do an advocacy action today because we need you. So advocacy, we're talking about policy and legislation to benefit the ALS community. We're doing this locally. We're doing it statewide in California and Hawaii, and also across this beautiful nation. We really focus on, you have our focus areas up there. We focus on access issues, things where there are policy problems that are creating barriers to access to key medical, and also this includes equipment, funding for research, and of course, generally raising ALS awareness across our legislative bodies. We know that awareness is often the first step, that familiarity breeds affinity. We want people to know more about ALS so that when we have an ask, they will say yes. Related to that, I'm going to invite all of you to find an example of an ALS network action opportunity. If you find us on social media, ALS Network, or sometimes we're listed as your ALS Network. Just last week, we put out an action alert that enables anybody to go in, you put in some information, it will automatically identify your representatives, provide a template letter that you can customize, and then you push send and it will kick it off to those representatives. The action alert we sent out last week was specifically about the NIH cuts that are proposed that are absolutely devastating to the research ecosystem in the nation, not only for ALS. So if you want to if you've been feeling like I want to do something, research is so important, how can I help? There's a nice, easy way you can send a letter on that very important issue. So let's talk a little bit about research. We're really, as a patient advocacy organization, as the ALS Network, we are focused on supporting every person with ALS who's in front of us today, making sure they have the very best of everything that's available to them, and also taking action to help prevent ALS tomorrow, right? And this means accelerating the search for effective prevention strategies, treatments, and cures. And you heard from the presenters already today that we're not there yet. We're working on it, we're getting there, but we have a lot more work to be done. So how are we accelerating this search as a patient advocacy organization? Through a few different things. First, thought leadership. We need the best ideas, the best minds coming together. We need Don and Justin sitting next to each other and duking it out and arm wrestling. And Don is known for asking very difficult questions. You have a little bit of yes, you do have a little bit of a reputation for that. It's good. It's pushing the field forward. So every year we host the ALS Network Research Summit, bringing together about 250 researchers. It was very lovely to have iXCells here with us this January. And again, putting out what's happening in the field, what's working, what's not working, how can we learn, how can we keep moving things forward. We do also have, related to thought leadership and really encouraging thought leadership, the Fred Fisher Excellence in Research Award is a new research award that we're launching. We plan to announce the first recipients in next January to really help inspire others to be mavericks in their thinking, to take good risks. And then recruitment. So again, your patient advocacy organizations and the ALS Network, when it comes to ALS, we take research recruitment very, very seriously. We would like our researchers, our biotech companies, our pharma companies really focused on science, and we can be a great asset and are a great asset, to helping to populate that research that you're doing and get it fully populated faster so that we can get things going. And we also help populate other research opportunities. It's not only clinical trials. There are a lot of other ways the ALS community is participating in research, including, if you don't already know this, there is a national ALS registry that's hosted by the CDC too. So then funding research, I'm going to talk a little bit more about that momentarily, but another way the ALS network is contributing and accelerating research is by directly investing in it. And we take this very seriously and are proud to do it. And then collaboration. It's going to take all of us. It's a very complicated disease, and we're happy to be collaborating to help accelerate, act as a catalyst to our friends doing this research. We have a world-class scientific advisory committee. 2 of the members might be sitting in front of me, who you heard from earlier. And again, we're happy to collaborate with them and have their input and great thinking informing the research that we're funding. We also co-fund research. We're very open to that and love doing it. We co-fund research with ALS United. We're also proud to be a member of ALS United, and it's exciting to be pooling dollars to propel research. We co-fund research with the ALS Cure Project and also with Target ALS. So again, this example of how patient advocacy organizations and nonprofits can pool their dollars to really push good science forward faster. We're at the table for that and oftentimes leading that. So this ties to partnering with industry, and it's important and we're happy to be doing it. How do we do this? How do we go about this? So the first part I mentioned, directly funding research. So we're looking at high risk, high reward, smallish grants. ALS Network in late April, we're launching our first request for proposals, and we will be funding 7 innovation projects at about $150,000 each. It's open to anybody in the United States. So anyone, anywhere, if you have an innovative ALS idea up your sleeve, please submit an application this spring and put your hat in the ring. The other thing that we're doing, I'm going to mention one other thing about our direct funding and that request for proposals opportunity. Something that we're also being really aware of, and again, this goes back to the urgency concept, is okay, we want to directly fund research. It's got to be great. I'm not a scientist, so these guys are going to figure out, and the 22 other people on the scientific advisory committee, they're going to decide which are the best ideas that should be funded. But how do we do that quickly? So we're working within a very nimble approach. It's a brief application followed by the people who are invited, make it through the paper application screening, are invited for a fast pitch session. And we're doing this fast pitch with the reviewers. They're face-to-face. We know scientists oftentimes don't get an opportunity to interact with reviewers at all. They'll be able to interact, Q&A, right then and there. The applicants jump off, and then the scientific advisory committee makes a go, no-go decision pretty quickly. And then we will rapidly move from there into contracting, and then we'll plan to issue the funding in the same year as the application is submitted. So again, how can we be additive in this research space and bridge gaps and do it differently? And I want to also mention that you can be a biotech company. You can be for profit. You can apply for funding with us, even if you're a for-profit entity. Again, we want to fund the best science. And in fact, one of the projects we're funding this year ties to Don Cleveland's presentation, where there may be an alpha-synuclein subtype in ALS. So we're funding an innovative company named Amprion, who's looking into that to confirm if there may be a subtype in sporadic ALS related to alpha-synuclein. So then advocacy, education, and consulting ties to community-centric approaches. We are there. We have a community research committee made up of people from the ALS community, people with ALS, caregivers, direct lived experience. And these are folks who can interact with our research partners, who can come in and make sure, is your trial design community-centric? What does a person with ALS think about what you, researcher, scientist, are planning to do? And are you on the same page? We can bring that perspective into the room, and we're happy to do that. And a lot of the advocacy I mentioned, education as well. More than once, I've come into a biotech company or a pharma company and provided a presentation or been on a panel about what is patient advocacy and why does it matter? And just really making sure to help the scientific community understand that. That's a service that we can provide here at ALS Network. We also have found ourselves, we're pretty good matchmakers. We have connected scientists. We're talking to so many of you, and we'll talk to one over here is doing this thing, and we'll realize you need to talk to this one over here. And so let's put you together, make the magic. Okay. And now it's up to the rest of you. And there have been some really powerful things that have happened because of that. So we have fun doing that, and also help, again, propel innovative ideas by doing that. And then we also partner with industry as a way for industry to give back, and a way for industry to be in the community that they're working so hard for. And one of the things I learned that I was surprised to learn early on is that there were a lot of scientists in labs who never met someone from the community. So then we have an opportunity to make that different. And there are a lot of companies who have the ethos, who want to make that different. And they're the ones who we see at our community events. I'm going to talk a little bit more about that in a moment. So one way everyone here who's from California can make a difference this year, if you want, I know you all want, like, who wants to help directly fund ALS research? I mean, everybody wants to, right? Everybody wants to do that. ALS can affect anyone, so it's on all of us to be a part of the solution, right? Here's how you can do it in a really, really easy way. If you're in California and you are filing your income tax form, on that voluntary contribution part towards the end, don't skip over it. Stop there. And we're the newest on the list, which means we're last. Not my favorite spot to be in. I'm going to be honest. I was hoping it was work the other way. Newest at the top. Scroll all the way down. We're code, it's line, but they use the word code 447. It's the California ALS Research Network is what you're going to hear. California ALS Research Network. 100% of those dollars will be reinvested in ALS research in the state. So I encourage you all to donate any amount, and to encourage your circle to also donate any amount. So one easy way you can make a difference. And then you can also get involved in, Michelle talked a little bit about this morning. I think it was Michelle. Yeah. Talked a little bit about this morning. Join us in California or Hawaii at an event. We have so many different things that we're up to. And we have rides and we have walks and golf tournaments and, you can hike and you can do whatever. We have people who paddle across oceans to do this. We have a gala, all sorts of beautiful things. There's something for everyone. The other thing is you can be creative about how you're going to interact with the patient advocacy organizations you're connected with. And I love the example of iXCells, who participated in the Death Valley ride. And that is a way to get involved. We were a part of that as well. Denali has come up a couple of times in presentations. Denali and Cytokinetics, that each year they host an executive splash where they set up dunk tanks and people can pay to throw the thing and dunk the CEO. And that's a fundraiser and a fun way of getting involved. So we'd love to see everyone here involved. If ALS isn't your thing and ALS Network isn't your thing, that's okay. There's a patient advocacy organization connected to what you're up to, and absolutely being involved in supporting their work. They're supporting your work. We're supporting your work. So you can support the work as well, and also be in the community, right? It's the people with ALS who are making so much of the research possible once it gets out of the lab. So there's also that thing, how do we give back to them? And it really is by being involved. It really is, in whatever way that's meaningful for you. Okay, that's everything that I have. And I just want to say thank you for your work. And thank you for this session, and for our ALS researchers here. Thank you for choosing ALS. Thank you for choosing rare diseases. It's not easy work, and we need each and every one of you. So thank you so very much.
Yeah, related to Sherry's talk, we also have a small fundraising box here. So snacks. We have we try to get people involved in internal team, but compared to what you have achieved, this like sounds like a joke. But nothing is too small, right? So we are having our next speaker. This speaker is dying from a different country, China. So this this event is representing a really global thing, right? So in the morning we have people dying from Poland. Now it's the 8:30 something in China. So in the morning, so we have Longchen Li, a physician scientist dedicated to translating basic research discoveries into transformative therapies. With over 20 years of research experiences and 13 years of clinical practice, he co-founded Rectigen Therapeutics in 2016 to advance his pioneering work on RNA-mediated gene activation into clinical trial. He is a former associate professor at UCSF and professor at Peking Union Medical College Hospital. So I visited his company in Suzhou, China, just last summer with Dong. We were very, very impressed by his innovative delivery and RNA targeting platforms. When I first saw his SOD1 mouse survival data, I almost couldn't believe it, because it's too good to be true. It's 200% more effective compared to tofersen. So I went there with Dong in the morning. We spent 5 to 6 hours, very intensive discussion, throwing very difficult questions. But at the end of the day, I believe I was convinced this is such an innovative delivery system, and also the new modalities. So we hear a lot about antisense. You will also hear a little bit antisense, but it's in a different way to treat ALS. So without further ado, I would like introduce Longchen to introduce his new delivery, RNA therapeutic delivery platform called SCAD.
Okay, give me one moment. I'm sharing my screen. Can you see my screen? Hello? Can you hear me?
Yes, we can see your screen.
You are ready to go,
great.
Dr. Lee.
Okay. Thank you very much. And then wait for your invitation and for your last introduction. It's my honor to join this event from let me see. It's not advancing from the beautiful city of Suzhou here. Okay, everything good. Rockgen is a clinical stage multi-platform company founded in 2017 on the siRNA modality or technology. A few years ago, I think 4 years ago, we thought we have the technology, we can deliver duplex RNA to the CNS. Why don't we tackle ALS with our technology? So obviously, the first target we want to work on is a sort of one, and very quickly, this program advanced from a concept, from idea to IND enabling, and to patient within just less than 3 years. So of course, this success was built on the foundation of what Don has done in this field, and also the many other work have done by in the Anderson's oligo field. And to build on the success of the SOD1 program, we quickly launched another program targeting fast ALS, and this program is now also in, we have reached the PCC stage, waiting for advancing this program to IND enabling. And of course, the major focus of our work still is on saRNA. So we have a saRNA program now in phase 1 trial, which is an oncology program. We have another saRNA program. We use saRNA to turn up a gene for the treatment of DMD, and this program is now in IND enabling. As you can see, Rockgen is really focused on a lot of rare disease. So today I'm going to introduce our SCAD delivery platform, which is enabling several CNS programs, and focus more on the ROC17 program for treating ALS associated with SOD1. So oligonucleotides fall into two major categories, single-strand ASO and duplex RNA, including siRNA and saRNA, which is what we are working on as our main technology. The key difference arises from the backbone modification. So ASOs use uniform phosphorothioate backbone or PS modification, and this promotes strong plasma binding, protein binding, leading to broad tissue distribution and visual in vivo activity without specialized delivery. So in contrast, different can only have limited pH modification, their function, which restrict the bioavailability. As a result, effective delivery systems are required for therapeutic efficacy, particularly outside the liver. So currently, FDA approved the siRNA drug designed to for liver target delivery using platform like LNP and the GalNAc. However, delivery siRNA to extrahepatic tissues, especially the CNS, remain a significant challenge for siRNA. So this situation underscores the critical need for innovative delivery platform to enable effective CNS delivery of siRNA therapeutics. So we know that siRNA is generally more potent than gapmer ASO for gene knockdown, as demonstrated in this example. So the two siRNA targeting sort of one gene were almost 100 times more potent than the gapmer ASO targeting the same gene in culture cells. And this ASO use the in this comparison match this sequence in chemical modification, not toxin. So the key challenge now lies in how we could translate this superior in vitro potency of siRNA into effective in vivo activity. To address this issue, at Rockgen, we developed the SCAD, which means Smart Chemistry Aided Delivery, and it's a simple and efficient delivery system. SCAD leverages the self-delivery property of ASLs through a unique design that links a single-stranded oligonucleotide, which we call accessory oligo or ACO, to one of the strands of the RNA payload. Its modular architecture allows independent optimization of the payload and the delivery component. When administered locally to confined space like the CNS, non-SCAD achieve broad biodistribution and long-lasting activity. It's compatible with both siRNA and saRNA delivery. So for the CNS, SCAD enable RNA delivery via intrathecal injection with no dosing frequency. Potentially every 3 to 4 months, once every 3 to 4 months. And this technology is undergoing clinical validation and advancing multiple CNS program at Rockgen Therapeutic. So the process of creating a SCAD-enabled drug candidate is very straightforward. You can first off your duplex sequence in vitro with chemical modification, and then conjugate one of the strand with the ACO. And these graphs show the scarification process and how it retained very well the potency of the siRNAs, highlighting the benefit of modular design. And of course, it's also possible to start with fully chemically modified duplex and the scarified duplex at the design and the screen stage. To evaluate SCAD siRNA in vivo, RNA was administered in mice via ICV injection at 20 nanomole. So after 14 days, it achieved over 80% SOD1 mRNA lockdown across the CNS regions, including the spinal cord, indicating broad diffusion, while peripheral tissue like the liver show minimal activity. So in comparison, the same siRNA without the delivery component exhibited only marginal activity. So we did directly visualize the distribution of Cy3 labeled as scattered siRNA after ICV injection. 3 days after injection, we could see broad and uniform Cy3 signals throughout the CNS, including the brain and the spinal cord. So we also evaluated biodistribution of scattered siRNA in rat by in situ hybridization. 14 days after single intrathecal dose, the Anderson strand of the dual-class was detected in different parts of the CNS, including spinal cord, as shown here as an example. So next, we wish to know how long scattered siRNA tissue exposure and its lockdown activity could last. So mice were sacrificed at the various time point up to 5 months after single ICV dose. In the motor cortex, which is near the injection site, the drug concentration peaked immediately, here as shown here, followed by a slow decline with substantial level remained at 5 months. So here, the red line represent the SCAD conjugated siRNA. The blue line is the siRNA, the same sequence without the delivery component. So you can see the duplex along also showed high and durable drug exposure in the motor cortex. So now let's look at the PD activity. So both the siRNA and the scattered siRNA peaked at the 2 weeks, but the scattered siRNA demonstrated significant deeper and more sustained lockdown. So despite the high drug exposure, siRNA lockdown activity rapidly returned to baseline. So this is the site near the injection. Let's look at the deep brain structure. The substantial higher drug concentration was achieved by the scattered siRNA with peak lockdown activity at 80%, which sustained very well towards the end of the study. At the most distant part of the CNS from the injection site, drug concentration was lower overall than in the brain, but still detectable at the 5 months. So despite the lower drug exposure, peak activity was still reached over 80%, and residual activity lasted till the end of the study. So in the liver, there was an initial high drug exposure, which declined very quickly to undetectable. Similarly, there was a initial moderate PD activity, which returned to baseline in 3 months. Due to slower CSF turnover in large animals, such as monkeys and humans, so the durability observed in mice may translate to even longer lasting effect in monkeys and in patients. So indeed, in the CNS of a crop eating monkey, after 2 biweekly doses of scattered siRNA, over 80% peak mRNA lockdown was seen in both high and medium dose groups. And this activity was very well maintained over the 70-day observation period. So interestingly, CSF SOD1 protein level continued to decline at the last time point, suggesting a long-lasting PD activity of scattered siRNA. So we anticipate that even longer durability could be seen in patients. So most of the data I present so far have focused on the SOD1 gene. But SCAD technology is broadly applicable to and can target any gene with a duplex sequence. So here, a scattered siRNA targeting the tau gene was tested in PS19 mice, a model of a tau path. 2 IV dose of 10 nanomole administered on post-later days 179 and 254 significantly reduced phosphorylated tau protein accumulation in the hippocampus and the cortex to an extent close to similar to the wild type animal. So this demonstrates gut ability and the delivery siRNA to deep brain structure effectively, preventing or clearing toxic protein buildup. So next, I would like to introduce our ROC17 program powered by the SCAD platform. So ROC17 is a siRNA targeting the SOD1 gene aiming at treating ALS associated with SOD1 mutation. And this drug is administered to patients via intrathecal injection. And this program has received IND clearance from both FDA and the Chinese counterpart. IIT study has been completed with 6 patients treated, and a formal phase 1 study is currently ongoing. So it's widely accepted and the clinically validated concept that mutations in the SOD1 gene leads to toxic gain of a function. The ROC17 works by degrading SOD1 mRNA, reducing the synthesis of both mutated and the wild type SOD1 protein. And this approach helps to decrease the accumulation of toxic aggregates associated with the disease. In several preclinical studies using the SOD1 G93A model, our scattered siRNA has demonstrated remarkable therapeutic efficacy. For example, in this single dose efficacy study, the model mice were given 1 single ICV dose at PND 60 compared to the vehicle control. Scattered siRNA significantly sustained body weight and extend animal survival in a very nice dose-dependent manner. So similarly in rat, SOD1 G93 ALS model, scattered siRNA treatment also significantly improved the muscle strength and extended survival. Here we use a control duplex with the same SCAD architecture as a negative control. Here, the blue line, the red line is our ROC17 treated rat, which has similar performance as the white type rat, as indicated by the green line. At the end of the study, the remaining rat simply didn't die, so we determined to terminate the study because we already observed significant benefit of the treatment. As we know that the SOD1 G93 mice is one of the most used ALS model for studying therapeutic efficacy, not just for SOD1 ALS, but also for sporadic ALS. And this model typically shows signs of disease onset around the PND 100 days and reaches terminal stage around the PND 160. So in the literature, in order to demonstrate the efficacy, treatment is typically given before disease onset. So to our best knowledge, no one has ever tried to initiate treatment after PND 90. So encouraged by the remarkable treatment efficacy demonstrated in mice and rats study I've just shown, we decided to challenge the status quo and treat the mice way after disease onset, which is more relevant to clinical situation. So we give 2 doses of our drug, PND 126 and 151. And we also include a lot of early treatment group as a positive control. So reproducibly, early treatment demonstrate remarkable benefit in improving muscle function and extending survival, as indicated by the blue lines. As we anticipate, late treatment also shows significant efficacy in both improving muscle function and extending survival, as denoted by the purple lines. So this result give us a huge confidence that our drug could translate into clinical meaningful benefit in patients who have already developed symptoms. Building on the strong preclinical effects data, we initiated investigator-initiated trial or IIT in April 2023. And this is a multiple ascending dose study with the primary endpoint being the safety, focus on safety and the ALS function rating score, and the secondary endpoint including CSF31 and plasma NFL concentration. In this study, 6 patients have been treated with the longest treatment duration reaching 40 months. All the participants have completed the stage 1 dose escalation phase, with some now on stage 2 maintenance dosing. So I'm sorry today I can only present the interim result from this study. Because the final study report is planned for the 2025 AAN meeting in April. And these slides provide an overview of the dose regimen for all 6 patients at the end of the study. For the first 3 patients, the starting dose was 60 milligram per dose. So after 3 loading dose, the patient transitioned to bi-monthly maintenance dose. After seeing very favorable safety signal from the first 3 patients, the starting dose for the last 3 patients was increased to 90 milligram per dose. So after 2 loading dose, the patient transitioned to maintenance dose every 2 or 2.5 months. The highest dose reached for the first patient is 180, and for the rest of the 5 patients is 150. The interim safety data indicate that ROC17 is safe and very well tolerated. All the AE reported were mild, including muscle tremor and headache, with no DLT, no SAE observed. Notably, the AEs occurs only after the first few doses. And in later doses, patients reported no AE at all. And this is very encouraging that this drug is very safe, even at when it was escalated to a very high dose. So interim biomarker data indicate that the ROC17 treatment significantly lower CSF31 protein level, reaching 50% reduction after 4 doses. The ROC17 treatment also significantly lower plasma NFL levels, reaching over 60% reduction after 4 doses. So very encouragingly, ROC17 treatment appears to have stabilized or even improved ALS functional reading score compared to baseline, suggesting potential clinical benefit. Similarly, ROX17 treatment appears to have a positive impact on patients' lung function, further supporting its potential therapeutic benefits. So building on the strong preclinical and patient data, we are expanding our CNS pipeline using the SCAD platform. So a second ALS program targeting FAST mutation, as I have mentioned at the beginning, is now progressing towards the IIT study in China. We also have programs targeting tau for AD and the siRNA program for spinal muscular dystrophy. So in summary, the SCAD platform is a very reliable, efficient delivery system for RNA therapeutics targeting the CNS. So ROX17 has shown to be safe and well tolerated at dose up to 180 milligram, with evidence of target engagement and reduced plasma NFL level and stabilized or improved disease progression. So lastly, SCAD enable rapid development of new RNA therapeutic for ALS and other rare CNS disease. Thank you. I'll stop here.
Great. We're gonna wrap up, but before that, we have a Q&A for the last section. So can I ask the last 4 speakers? Also, Dr. Lee, if you can stay online.
Yeah, of course.
Hello. Yeah, I have one quick question for all the speakers, especially my mentor Don. As you already mentioned, that 3 failed clinical trial disease use ASO. One's Tangsin 2, another one is the Huntington, a third one is the C9. They all belongs to polyQ disease. So is that a coincidence that ASO cannot treat the polyQ disease? Or
So I don't think so, but and I mean they're not all polyQ. One, they're all repeat diseases, but the ataxin one, I think was just it's a complete drug failure. The suppression of ataxin was extremely modest, and I would have so I have no that was run by Ionis Biogen, and I have no part to that. But I looked at the final data; they barely suppressed ataxin, so I think it was just a complete failure. And now with regard to what Dr. Lee just described, it may well be that the drugs chosen, the ASOs chosen, were just simply not effective enough. And this delivery method that you just heard from Ractogen, and I'm going to say it in a much, in a very, very direct way, they're gluing an ASO onto an siRNA. And they're now making a quite big drug. But the question, already the ASOs are 15 times the size of a typical drug and heavily negatively charged. But by using the pathway of uptake, the ASO uptake has really made those siRNAs more effective. So I think we should be very open-minded that maybe this is and delivery has always been a key issue. It remains a key issue for neurodegenerative disease. So and improvements should be welcomed. And there I think the failed Huntington trial, the failed C9 ORF72 trial, I think there's the team, the broad team does not agree on the basis for the failures. So, and I'm sure that the audience here is going to have their own views as well about why they failed. Certainly the drugs were not as active as you would have liked. And there was some evidence that could be interpreted as the drugs had their own toxicities. And so dose dependent, that is indeed what Roche concluded in terms of the redo of the Huntington trial, where they're starting way earlier, patients way earlier in disease, but also with a lower dose of the drug because they were afraid that actually that they were seeing drug-mediated toxicities. And these ASOs, actually, I think for the in lorem and for the other very rare disease examples, the issues about toxicity of those ASOs, those are very real. The Ionis examples that I'm familiar with, they've made and tested 1,000 different ones every time they went to trial. Their competitors have frequently shown that they do know how to make toxic ASOs. And Ionis was kind of quite pleased that actually, and I should have mentioned in my talk, and then I'm going to shut up. So I meant to mention, especially because representatives of ALS Network were here today, the founding ASO that went into the human nervous system, Ionis was not committed to that project at the time. Their CEO was opposed to working in the nervous system, and the preclinical toxicology of the first drug that went into man was not paid for by Ionis. It was paid for by ALS Association, Golden West ALS, and by MDA. It was Tim Miller and I wrote applications to both of those organizations to provide $1 million of funding, a large amount of funding from each of those organizations in order to enable the first in-man trial. And because that was safe, that then enabled, that was the birth of the SMA program. It was the birth of Ionis's focus on neurodegenerative disease because they really could deliver them broadly and safely. But they work very hard to find ones that are sufficiently safe.
I might just add that in the case of C9, I think that the disease mechanism is more complicated than some people thought originally, and I don't think there was a lot of like listening to the cells and what the cells say about when you treat with a certain approach, which I think is a good lesson for just a lot of the things that we're doing now with these, N of 1 studies or other, ASO studies. And it's really important to test that therapeutic mechanism and make sure that's the right one.
So thanks, everyone. This has been really inspiring to think about. Looking at the heterogeneity and all the preclinical models that you guys have all presented and talked about, there have been a couple paths people have chosen when looking at these CRISPR mediated, so you have an isogenic control versus patient derived, so you can look across a panel. Kind of tying this to oncology in a multiple hit model. So you need some sort of genetic, then you need 2 or 3 other hits along the path to actually get disease. Do you feel like in these neurodegenerative settings, we need to be dealing with panels of patient-derived as opposed to 1 or 2 well-characterized models where we've CRISPR engineered in a diverse set of mutants?
So certainly many definitely think that, and I have to say I'm not one of the primary advocates that you need 1,000 in order to be able to get an answer. I'm actually worried about a different thing, and I'll let the others talk to that question, but then I add to your question, which is that and I know this is going to be deeply unpopular at iXCells. So the idea we had demonstrated, at least for disease in mice, that SOD1, it's a ubiquitously expressed gene, and the cells that drive rapid disease advance are not the motor neurons. They're the microglia and the astrocytes, their synthesis of mutant SOD1. So now if you're taking an isolated motor neuron, is that going to be adequate for, are we reverting to now assuming that the disease is cell autonomous from mutant action within individual cells? To me, that's actually, it seems to me to be unlikely to be true. Didn't say it doesn't play an important role, the synthesis within the motor neurons, but it does kind of, so my question to Justin or others would be, how are we going to, are we going to consider other contributions from those mutants in other cell types other than the primary motor neuron?
Absolutely, we're going to have to.
Yeah, I think that's a huge part of it. It's way more complicated to incorporate those cells in my experience, but it's really important. I think, for things like ALS, there's an opportunity where certain patients may harbor genetic lesions and genes that are highly associated with ALS, but maybe not. There's not a strong example of many families, where this mutation is separating with disease, but likely it may play a role in disease. And I think there are many people initiating efforts to make ASOs for those genes, and in hopes that it might help the patients that are just a subset of ALS patients with mutations in certain associated genes. And so there, I think it could be useful to see if indeed, this ASO strategy could work for those patients. This would just be a way of trying to get at the sporadic population in some subsets.
Thanks. I am as someone who is not a neuroscientist, but has been following the literature closely, and who thinks about this in terms of developing technology. I don't think that we know the answer to the question, but one reason that I find the work that Justin has done, and others like Alyssa Coyne and Joe Fauci in particular, that we now have several independent examples of these heterogeneous patterns showing up in the transcriptome, and I think that a big question that we need to answer is how much can we learn from that, and that is why with Project Mosaic, at least, we in the early phases, we want to know are are those patterns reproducible in the same mother neurons, and if so, is that indicative of the underlying biology? And and then if it is, we can begin to answer more complex questions around well how much can these models themselves about 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 how drugs might work in in different subgroups, and to your point, what can't these 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 models tell us? Sorry, and then that is where we will probably need more sophisticated sophisticated co-culture and eventually possibly 3D organize, but we have to start somewhere.
My question is for Justin. So I am very impressed by the results you showed that you did RNA CRISPR-I library screening to identify two target genes, which knocking down both either of them showed a protective effect. So I'm just wondering if you have gone to the other direction, so try to screen like overexpression library to see whether overexpression certain targets also may have a protective effect.
Yeah, thanks for that question. We didn't directly do that, but we do have genes that are sort of negatively enriched when you knock them down. So they sort of kill the cells more when you knock them down, and so those might be candidates, for overexpression. If maybe some of this RNA, small saRNA technology, can work to upregulate genes, I think that would be powerful if that could work. And so I'm kind of curious to see how that develops. Yeah.
Yeah, maybe if I can return to the former question as well. So maybe where the broad large number of samples, one place there are about 30 diseases for which there's a nucleotide repeat that's causative. Maybe the most famous one is Huntington's disease, but also for C9orf72 in ALS. And there, the different populations, right? The C9orf72 expansions are almost absent from China, but they are the largest cause outside of China. They're the largest known genetic cause worldwide. And there, there was a long discussion at the nucleotide repeat meeting about the partial penetrance of the C9orf72 mutations, with multiple authors identifying in different populations that it's only about 25% penetrant. So if you inherit that, the expansion, you have a real chance, but a 25% chance of developing disease and a 75% chance of not developing by age 80 in any ALS-like phenotype. And so there, maybe, so maybe I'd even turn that into a question for Justin or others. The question, if you do look at those, are there sequence variants that can now be protective? And is it in the in vitro making these neurons? Do we see differences in the phenotype in those?
Yeah, that's a great question. I don't think we've done enough lines carefully enough to say if there are differences. I think there's that TMM106B gene that might be a good example of with a certain variant in that gene, you strongly protect against getting FTD with the C9 repeat expansion. Maybe there are things like that for the ALS side. Yeah, I totally agree.
So that's definitely an area where many different cell lines, especially if they were collected like in ways in where they're really comparable, been produced comparably. Maybe that actually, I would also then I would really strongly advocate that that none of us should be arrogant enough and to think that we know what the right way to do it is, and nor should we think that the perfect should not be the enemy of the good. And so the strategies Justin now has proposed 2 or 3 targets. Those are good. Those are good targets. I proposed one today as a Stathman, where I think we can do good. It's not by no means perfect. It's not putting TDP43 back, but it's putting back one of the proteins that's required for maintaining innervation. And so this is and there'll be others. And so I would really broadly support the idea that different ideas are the right direction, and don't let Sherry's gonna kind of dislike part of this. The idea that organizations want to band together to identify the best idea that a whole broad spectrum of folks will think is an okay idea, that's probably not the right way to go. You want to see that the discoveries, the true discoveries, are going to come from unexpected directions, because otherwise they wouldn't be the grand new discoveries. So I would really want to see that a broad spectrum of folks that can defend their strategy. It won't be perfect, but it will be. It's another shot, and we need as many of these shots on goal.
I just want to say that those are the exact ideas we hope will come into apply, and that people like you with running labs who have scientists who may have a real out of the box idea, this RFP gives them a place to get some funding for that. And you're on the committee to help say, hey.
So again, while today's meeting, and I could see it's now just accepted as an obvious fact, the ASOs were initially not at all accepted, including at Ionis. The idea that they could work in the nervous system, that was not believed inside Ionis, and the investment was made by ALSA and then MDA to develop that, we did not understand that we could deliver these things broadly. We did not think we could. We thought we'd had to use continuous flow. We had no idea we could get 4-month efficacy periods, and as you saw from Ractogen, even better for this fusion of an ASO onto an siRNA. It's just like you have to do it and then critically assess it, of course. But then you applaud success. You're disappointed by failure. But that I think is the real process that has enabled us to get to ASOs are going to be used broadly in the nervous system. We heard it all day, right? I didn't include today, I do frequently, but I didn't today, the original, we sent that work for publication, including showing that it was effective in non-human primates. And that was editorially rejected as no contribution by Nature Medicine. It's like, then Huntington, we showed we could get partial disease reversal in 3 Huntington model mice. And they said, well, that was nothing either. It's just, and in the end, I got that editor on the phone, and he said, well, he admitted that showing that you could do it in non-human primates, it was remarkable. But then he concluded that it was no intellectual contribution. Because you knew if you turned the gene off, it would be good. And then he said, ASOs will never be used in human therapy.
We're running a little bit late, but I have one last question. Sorry, I took the last one. So for Dr. Lee, are you still there online with us?
Yes.
Okay. So because after hearing your presentation, I am again confirming my belief that the technology can be, because it's targeting, they have very tissue specificity, and also the delivery efficiency is much higher with this, conjugation with ASO. So I think this modality can be very beneficial to some of the ultra-rare neural disorders. So as one of the biggest audience today. So I'm just curious, do you have any strategy how to utilize your technology to help with those ultra-rare patients? I know we have a little bit discussion with Amber-free, SL661, but it seems that we don't have very clear roadmap and path how to move this forward. Partially it's because of the politic barrier, and also part of it because of the financial barrier. So I'd like to hear about your thoughts.
Oh, yes, of course. That's a good question. The last year at the Boston CNS delivery meeting, I talked to some people from the ultra-rare disease organization like Everyone Medicine, and so I proposed to them that if they are interested in using siRNA for ultra-rare disease, and they can use the scattered delivery system to quickly deploy the therapeutic, because it's just like a plug and play, right? Very quickly. And actually, we are doing this in collaboration with the very famous patient, ALS patient, China, in China. We are now designing and the screening allele-specific siRNA for some mutation identified in a certain patient. Yeah, it's quite feasible.
All right, so I guess we are closing. We're gonna have a closing remark by our one of the speakers. So can I ask? Thank you so much for you guys. So I ask you to stay with us a little bit, 5 more minutes. As a tradition of our events, we always think this kind of events and initiatives should be patient focused. So the patient's voice matter a lot. So we always ask one of our patient advocates to close our events. So this year, so it's gonna be Michelle.
Thank you so much. I just want to take a minute to highlight the need right now for cross-disease collaboration, especially right now. It's obviously an extremely uncertain time for the rare disease space, both in terms of federal funding and potential changes at CIRM, as well as ongoing challenges on the industry side. So it's really, and we all know that when funding dries up, rare disease programs and grants are the first thing to get cut. So it's really important for us to all come together and put a spotlight on rare diseases and bring attention to the needs and the opportunities in this space. We need to get the message out in the mainstream that rare is not actually rare, bring attention to these devastating diseases, but also the historic and transformative treatments that are on the horizon. And I'm going to put in a shameless plug for my Zebra Run for Rare, which is not actually planned yet because the zoo is not cooperating very well. But I'm not letting the dream die. As one of the world's leading biotech hubs and the home of the world famous San Diego Zoo with our real life rare disease mascots, I think San Diego is uniquely positioned to host a really large scale, high profile event that can raise funds and awareness for rare diseases. Nyanwe and I and some other groups were actually in talks with the zoo last year, and we even had the mayor's office involved, but in the end they kind of told us that, well, we can't do any more runs unless it's for organizations with close ties to our board. So if any of you happen to have high-level connections at the zoo, please let me know. Oh, there's also going to be a great event on Rare Disease Day downtown at the Brigantine next week. So it would be great to see many of you there to continue these connections and collaborations. Finally, I just want to say thank you to iXCells for hosting this event. Thank you to everyone and all of you online as well for being here today. Thank you all for caring about rare. I have 100% confidence that life is not always going to be like this for my son Tristan and for our family. And I know that by working together, collaborating to accelerate progress, we can make a difference for hundreds or thousands or millions of other kids and families like mine. So thank you all.